this is Dr. Priyadarshini, one of the mentors with StudyMRCOG, and today we are going to discuss about embryology. Going to the stages of development, we have fertilization followed by cleavage, caspillation and organogenesis, which corresponds to embryogenesis, followed by maturation. In fertilization, what happens? We have sperm which is going to enter into the ovum, and there's fusion of the egg and sperm to form the zygote. So what happens? The fertilization takes place in the ampullary region of the fallopian tube, and then the division starts. We have the 2 cells which is the zygote, then we have the 4 cell and the 8 cell which slowly traverses the fallopian tube to reach the uterine cavity. So day 1 we just have fertilization taking place. In day 2 we have cleavage. Next there is compaction and differentiation. By day 5 there is cavitation leading to a blastocelic cavity. We have the inner cell mass and the outer cell mass. The outer cell mass are the trophoblast which go on to make the placenta, and the inner cell mass is the embryogenic mass which is going to go on to make the embryo per se. After that, the hatching takes place where the zonar layer disappears, and there is by day 7 we see implantation. After implantation, there is cell mass differentiation which is noticed by about day 9, and we have the differentiation into epiblast, hypoblast, and we have the trophoblast. This is the bilaminar disc formation that takes place. After the bilaminar disc formation, we have formation of mesoderm leading to the trilaminar disc formation. So in the bilaminar embryonic disc we have the epiblast and the hypoblast, whereas in the trilaminar disc we have the ectoderm, mesoderm and the endoderm. Correspondingly, when the trilaminar disc forms, we also have the beginning of organogenesis taking place. Now it is important to know about the derivatives of each of these layers. Now as I discussed, we have ectoderm, endoderm and the mesoderm. Ectoderm is the outermost layer where we have the formation of epidermis, that is the skin, hair, the mammary glands, the epidermal placodes like the lens, the inner ear, the neural tube, the brain and the spinal cord and the peripheral nervous system form from the ectoderm. Coming to endoderm, it is where the yolk sac, the primitive gut, the lung, the liver, pancreas and the digestive tubes are going to form, and the mesoderm is going to give rise to a large number of structures, notably the axial mesoderm giving rise to notochord. We have the paraxial mesoderm giving rise to sclerotome, myotome and dermatome. From the sclerotome we have the axial skeletons, the myotome for the muscles, the dermatome is for the connective tissue of the skin. The intermediate mesoderm gives rise to the Mullerian duct and the mesonephros. The mesonephros is from where the kidney, the ovary and the testis is going to arise. The lateral plate mesoderm, we have the somatic and the visceral mesoderm. The somatic gives rise to the connective tissue of the body wall and the limbs, and the visceral mesoderm gives rise to the mesenteries, the heart and the blood vessels. Now these are the derivatives as we were discussing. In the midline we have the cauda mesoderm which is going to give rise to the notochord. Following that, next to it is the paraxial mesoderm, and laterally we have the lateral plate mesoderm. In between the paraxial and the lateral plate mesoderm we have the intermediate mesoderm. It is of importance to know this because these are some exam tested areas where they ask the derivatives from these mesoderm. Neurulation. Now neurulation is a process where the nervous system is going to develop. We see that initially we have the neural plate, and all these are derivatives from the epiderm. So we have the neural plate that is being formed, which is later seen as a groove. So that is called as the neural groove. And next there is deepening of the neural groove, and in the sides we see the somite formation. Finally, the neural tube, the tube forms after the neural groove, and it is from this neural tube we have the cranial end and the caudal end. The cranial end will go on to form the brain and the corresponding structures, and caudally we have the spinal cord formation. Coming to the cardiovascular system, the cardiovascular system, as we see, is mesodermal in origin, and the cardiogenic field is established just after gastrulation, that is around 18 to 19 days, and the fully functional multi-chambered heart is seen by the 8th week. Now, coming to the important summary of the development of heart, we have the various heart chambers. There are basically 5 different dilatations. They are the sinus venosus, the common atrium, the common ventricle, the bulbus cordis, and the truncus arteriosus. Now, once these dilatations are formed, there is rotation of these tubes to form the final structure of heart. The embryonic dilatation of the sinus venosus gives rise to the smooth part of the right atrium and the coronary sinus. The embryonic dilation of the common atrium gives rise to the rough part of the right atrium and the rough part of the left atrium. The common ventricle gives rise to the rough part of right ventricle and the left ventricle. The bulbus cordis is going to later on form the smooth part of the right ventricle, which is the infundibulum, and the smooth part of the left ventricle, which is the vestibule. The truncus arteriosus is the origin of the pulmonary trunk and the ascending aorta. Coming to the development of the respiratory system, we see by the beginning of 4 weeks, the pharynx, the laryngotracheal tube, is going to start forming, which later on leads to the tracheal buds, and these tracheal buds is going to break down further or buds further to form the bronchial buds, and the bronchial buds will further keep on dividing till up to 8 weeks to form the respective lobar segments, the right having upper, middle and the lower lobe, and the left having the upper and the lower lobe. So by 8 weeks we see the complete formation of the respiratory system. Foregut, midgut and the hindgut. If you see, the foregut is corresponding to the second part of duodenum. From the second part of duodenum up to the first 1/3 of the transverse colon forms the midgut, and after that is the hindgut. Going on with the urinary system. The urinary system is of importance because the urinary system and the genital system almost correspond to each other in development, and most of these congenital malformations, if it is seen in the urinary system, it is worthwhile to evaluate the genital system too, and vice versa. Now, the urinary system, if we take, we have initially the pronephros, the mesonephros and the metanephros. Now, out of these 3, the pronephros and the mesonephros usually degenerate, and it's the metanephros which is going to give the final structures of the adult. So the urinary system and the genital system are very closely associated. They both develop from the intermediate mesoderm. It is usually seen from the 7th to the 18th somite level from the 3rd week. The nephrogenic mass, which is called also the cord, it is seen in the dorsal side of the coelom. Each cord produces a bulge, and this is called as the urogenital ridge. Now, the urogenital ridge is the one which is going to give rise to the urinary and the genital structure. The nephrogenic tissue, which is going to form from the 7th to 14th somite, is going to break up into small segments, and these segments are called as nephrotomes. Now, the kidney, as I mentioned, is going to develop from the 4th week in 3 different steps. The first one is the pronephros, which forms and just atrophies. So there is no function for it as such. The second one is the mesonephros, which is again a transitory in origin, and finally the definitive kidneys are going to arise from the metanephros. Now these are, as I mentioned, is mesodermal in origin, and we have the urethral annulage, which is going to start its origin from the Wolffian duct. The main functional unit of the kidney, which is the nephron or the urine excretory part, is basically arising from the metanephric annulum. Okay. Okay, whereas the rest of the upper urinary tract, like the collecting ducts, the calyces, the renal pelvis and the ureter, they develop from the ureter unplugged. So coming to the lower urinary tract, the lower urinary tract is going to differentiate from the cloaca, and this is seen somewhere between the 5th to 8th weeks, and we have the cloaca which gets subdivided by the urorectal septum. The ventral part of this is going to form the primary urogenital sinus. Now from the primary urogenital sinus, we have development of the urethra in the lower part and bladder in the upper part. The urethra at large discharges into the upper posterior wall of the urogenital sinus. So basically we have two things which gives rise to. In males, the Wolffian duct is going to remain present and forms a connection between the genital tract in the lower part of the urogenital sinus. So this is what we were discussing. We have the pronephros, the mesonephros and the metanephros, and lower down we have the cloaca which gets separated by the urorectal ridge and form the urogenital sinus. Ureter is basically an outgrowth of the mesonephric duct, and the terminal ends of the mesonephric duct become part of the bladder wall. The ureter, as the time goes, it will have a separate entrance into the bladder. Now, coming to the development of the urinary bladder, as we were already seeing, the cloaca gets subdivided by around 4th to 7th week. The posterior part is going to be a part of the anorectal canal, and the anterior part, which is the primitive urogenital sinus, will give rise to the urinary bladder. It is basically from the upper and the largest part of the urogenital sinus which forms the bladder. Initially, if you see, the bladder is continuous with the allantois. Later on, the allantois gets obliterated and gets replaced by a structure which is called as the urachus, which actually forms as a connecting apex of the bladder with the umbilicus. In the adults, this urachus gets converted into the median umbilical ligaments. Now, if this is going to be patent, then we see urine being discharged from the the attachment of umbilical or the umbilical point in neonates, which means that the urachus is not completely obliterated. So bladder mainly from the vesical part of the urogenital sinus, and trigone. The trigone which is seen in the base of the bladder is absorbed from the caudal ends of the mesonephric ducts. The epithelium is endodermal in origin, and all the other layers are from the splanchnic mesoderm. So this is the division of the cloaca. We have the primary urogenital sinus and the anorectum. The primary urogenital sinus divides into the vesico-urethral canal, which makes the bladder, and in males it goes on to make the prostate and parts of the pars prostatica, membrica and spongiosa. Whereas the definitive urogenital sinus has the pelvic part and the phallic part. Now, the urethral plate also forms from the phallic part, and in females, part of urethra, part of vagina, labia minora and vestibulum is formed from this. This is the fate of the primary urogenital sinus, where we have the division into three different parts: the cranial or the vesical part, which is going to form the bladder; the middle part or the pelvic part, which is going to form the main portion of the male urethra; and the entire female urethra. The caudal part of this is called as the phallic part, which is going to grow towards the genital tubercle. Now, coming to the most important part, which is the development of the gonads. The gonads are derived from three different sources: the mesothelium, which is mesodermal epithelium, which lines the posterior abdominal wall. Second is the underlying mesenchyme, which is the embryonic connective tissue, and also the primordial germ cells. So what happens? These primordial germ cells, they arise from the yolk sac and they have to migrate towards the posterior abdominal wall. It is in the posterior abdominal wall where all the structures are going to be finally formed. Coming to sex determination, it is seen that before the 7th week, the gonads of both the sexes are identical in appearance, which are called as indifferent gonads. The development of male phenotype requires a Y chromosome. So this SRY gene for the testis determining factor has been localized in the sex determining region of the Y chromosome. So 2 X chromosomes are required for the development of female, and the Y chromosome, which has the testis determining effect on the medulla of the indifferent gonad. So any time where there is absence of the Y chromosome is going to result in the formation of ovary. Now, the testosterone that is produced by the fetal testis is going to determine the maleness of the fetus. Whereas the primary female sexual differentiation does not depend on the hormones. So even if the ovaries are absent, we can see there is a female sexual differentiation. Now coming to development of the testes and ovaries, as we said, by week 7 till week 7 it's going to be indifferent, and by week 7 it is going to start the differentiation, and by 12 weeks we are able to see the completion of this. After development of the gonads, we have development of the genital ducts, where we are going to see with the development of the internal genital organs and the external genital organs. Now coming to the internal genital organs, we have both the male and the female embryo have 2 pairs of genital ducts. What are they? They are the mesonephric duct or the Wolffian duct, which is going to have an important role in the development of the male reproductive system. Whereas if you take the paramesonephric duct, which is the Mullerian duct, they have an important role in the development of the female reproductive system. Now till the end of week 6, it is going to be an indifferentiation in the genital system. That is, both ducts will be present. So the dependence of this SRY gene and the determination of whether it's going to be a male or the female will determine which of these ducts are going to be present and which is going to be absent. Now this is how the differentiation or the development takes place in the males and females. Now in male, we know the predominant duct is the Wolffian duct, which leads to the development of the ductus epididymis, the vas deferens, the seminal vesicle, the ureter, the renal pelvis, calyces and the collecting ducts. The similarity which is seen in the females is the Wolffian duct is going to give rise to the ureter, the renal pelvis, calyces and the collecting ducts. However, the vestigial structure that is formed is the Gardner's duct. Now coming to the Mullerian duct, we see the importance is in the development of the female structures, where you have the uterus, the tubes or the fallopian tubes and part of the vagina. In males, it is going to form the appendix testis and the utriculus prostaticus. Now, as we were discussing earlier, the urogenital sinus is going to give rise to the bladder and urethra in both the sexes, whereas in male it gives rise to prostate and in female it forms the caudal part of vagina. Coming to the tubules of the mesonephros, as we were discussing earlier, the mesonephros usually disintegrate, but sometimes there can be small tubules and remnants, which in females can present as the epophuron and the paraphuron, and in males it can present as ductuli efferescens and paradigmus. Okay, so in males, we know the male external genitalia has the penis and the scrotum, and in the females we have the labia majora, the labia minora and the clitoris. So penis will correspond to the clitoris, the scrotum corresponds to labia majora and a part of the penile raphe corresponds to the labia minora. It is also important to know regarding the congenital anomalies of the female genital tract, which can either present in the questions as a picture or as an ultrasound image. So some congenital anomalies that we see normally are uterine didelphys, where we have two different horns of the uterus and we have two different cervix also. Arcuate is usually not considered as a congenital anomaly but just a structural deformity where there is more flattening of the fundus. Coming to unicornuate uterus, in unicornuate uterus there is only development of one of the horns and the other horn can either be completely not present, or there is no cavity, or there is a horn and cavity but it is not communicating, or there can be an underdeveloped horn with the cavity which is communicating with the well-developed horn. Coming to biconjugate uterus, in biconjugate uterus there is two different horns and there is incomplete fusion of both. So this can either lead to a partial or a complete biconjugate uterus. The difference between biconjugate and didelphys is that in biconjugate there is a single cervix and in didelphys there is a double cervix. Now coming to septate uterus, in septate uterus basically there is incomplete absorption of the septum or incomplete absorption of both the tubes after fusion. So depending on the level of septum, it can either be a partial septate uterus or a complete septate uterus. Now, coming to one more important aspect is the development of the pharyngeal arches and the pharyngeal clefts. Now, the pharyngeal arches and clefts are derivatives which we see near the buccopharyngeal membrane. The buccopharyngeal membrane disappears to form the future oral cavity. Now we have 6 different arches and 4 different pouches and clefts. Now what is the importance of this? For each arch we have separate derivatives like the skeletal derivative, the arteries, the nerves and also the muscles. Now coming to the pharyngeal folds, the fold 1 corresponds to the external auditory meatus, 2, 3, 4 corresponds to the neck. Going to the pouches, pouch 1 corresponds to the middle ear auditory tube, 2 to the supratonsillar fossa, 3 corresponds to the thymus, the parathyroid gland, and 4 again to thymus, parathyroid and the ultimobronchial body. Now coming to derivatives of the arches, they say arch 5 is usually disappearing. Arch 1 is the mandibular arch, the corresponding artery is the maxillary artery, the nerve being trigeminal nerve, and the muscle and the skeletal derivatives are the muscles of mastication along with skeletal derivatives like malleus, incus, the Meckel's cartilage. Arch 2 is the hyoid derivative. The artery is going to be the hyoid and the stapedial artery corresponds to the facial nerve and the muscles of facial expression. The skeletal derivatives being stapes, the styloid process, the stylohyoid and a part of hyoid cartilage. Third arch corresponds to the internal carotid artery and the cranial nerve being the glossopharyngeal nerve. The muscles are the stylopharyngeus and parts of the hyoid cartilage is the skeletal derivative. Arch 4 corresponds to the right subclavian artery and the aorta. They are considered as derivatives, and the nerve is the vagus nerve. The muscles are the pharyngeal and the laryngeal musculature, and the cartilage is the laryngeal. Now these are some very important aspects that has to be remembered for the preparation of the module embryology for the part 1 examinations. Hope this was useful. Thank you.